Thank you so much. I'm so honored to be here. Down syndrome is changing a lot in the last few years. Originally, we thought it was caused by one extra chromosome, number 21, which of course is true, and therefore it's got 100 extra copies of those genes on that chromosome. But people are moving to a new perspective, which is the brain development perspective. And treatments have been tried out in mice and found to be successful. Down syndrome mostly affects language, memory, locomotion. There's early dementia and, of course, mental retardation. In this talk, I want to think a lot about the process of neurogenesis, which is our neurons proliferate, and then we lose about half of them if they don't make good connections. If you see any child that has a small head, that's a child that had a problem with neurogenesis. And after the brain cells replicate, they have to connect to each other and talk to each other and exchange trophic molecules. To the extent that they do that, they survive, and to the extent that they don't, they're lost. So neurogenesis, the replication of the neurons, about 8 to 16 weeks, 18 to 25, they migrate. After 26, they land in the cerebral cortex, and the dendrites grow like trees. And at every step in the way, you make a lot more than you need, and then you lose a lot, like half. Also, to help this process along, there are certain projections, like the basal forebrain cholinergic neurons and locus coeruleus neurons, that they project all throughout the brain, and they give some innervation and support so that the cells that just landed there will have some sort of trophic support and prevent their early death. All right, so now let's think about brain development as a physiologically modifiable process. Let's say we had a normal brain with brain cells A, B, and C. Now, second brain number 2, we're going to augment neurogenesis, and we'll make more neurons, but we'll lose them. Does that leave us in the same place? Not necessarily. Maybe after we lose them, the ones that remain have better connections. So in brain number 2, you see AA and A. You might have the same number of neurons, but better connected. And so that might be a better brain. And on the other end, if this neurogenesis process doesn't work very well, then you may have limited neurons in a small head. And in our own personal experience, oftentimes we might grow up in a house where only one language is spoken. Every child has the ability to learn as many languages as it's exposed to. So a child that would be exposed to one language will learn one language, or a child that would be exposed to 3, 5, or whatever will learn that many languages. And conversely, where there's sensory deprivation, normally grown circuits will disappear. You might be perfectly biologically normal, but if you do not receive adequate stimulation, your capabilities will disappear. There's been a lot of research over the years about the nutrients in Down syndrome. In the beginning, where the brain is growing explosively, nutrients must be important. And in fact, the brain grows by functioning. So that means anything that affects brain function will also affect brain development. And also, from the development perspective, timing is everything. If you took folic acid or folate and you had that 28 days after conception, you could close your neural tube and you wouldn't get spina bifida. If that didn't happen, and the baby has spina bifida, it will require surgery at birth and permanent paralysis. At that time, no amount of folate could undo the damage. And so therefore, a key concept is the timing is even more important than the treatment, or the timing is everything. All treatments and insults are much more effective early, and good treatments would have little benefit if given too late. Much of the research that's been done on Down syndrome, it's been done on children over the age of 5 or even adults. Maybe those were effective treatments, but given too late to have any benefit. When I discuss these Down syndrome model mice, the scientists have tested them in every way. And I'm not going to burden you with all the details. We could just take it in some headings. When they're testing behavior, they're testing for learning deficits, maze performance, attention, hyperactivity, stuff like that. When they're testing physiology, they're looking at neurotrophic molecules, and they do electrophysiologic things to determine how brains learn. And when they're doing anatomy, they're counting the number of cells that survived and how the dendrites grew and stuff like that. And these are just all parts of the same elephant. Environmental enrichment can do amazing things. If we put a mouse in a cage and throw some toys and furniture and playmates in there, the development of that brain could change dramatically. Prenatal stimulation of the mother will cause progeny that are smarter when tested after birth. So the effect of a treatment could go way into the future and across delivery and generation. So various experiments have shown environmental enrichment. You know, for humans, that could be reading to your child, playing with your child, stuff like that. Improves current and future brain performance. Increased tolerance to current and future stress. Protects against current and future injuries, mitigates toxins, mitigates or rescues the effects of disease genes. And one of the interesting things about thinking of things from the developmental perspective is the treatments, it's not that important which one. The diseases, it's not that important which one. Okay, so now let's see what the scientists have been doing with the mouse model of Down syndrome. In general, all right, so they used only environmental enrichment and exercise. They improved the anatomy, remember cell counts and all that stuff, behavior and learning, maze and memory and deficits. So on this mouse model, you could substantially improve, or almost to the point of curing, with merely environmental enrichment. You could use choline. Choline is a methyl donor, so that would enhance the production of DNA, RNA, proteins, and neurotransmitters, but also it's a component of acetylcholine and a support for, remember I mentioned those early projections that support the entire brain and give afferent support while the cells are trying to connect with each other. Those are the basal forebrain cholinergic neurons. So giving choline to pregnant mice improved their cognitive function as adult mice. And there's another cholinergic drug, donepezil, it's used in Alzheimer's. It had relatively little effect, but the interesting thing was teenagers benefited more than adults. People have thought a lot about free radicals because there's a gene on chromosome 21, copper zinc superoxide dismutase, which has as its function to generate hydroxyl radicals. If one would give vitamin E from conception, one could make a big difference. And then many studies given later, relatively little effect. Serotonin agonists. The hippocampus is one of the areas of the brain that's very important in memory. And adults have continued neurogenesis. We still make brains in our hippocampus even in adulthood. That's increased by serotonin input, and patients with Down syndrome are depressed. So you could ask, and people have tested, if we gave serotonin agonists, would that increase the neurogenesis of the hippocampus and memory? And the answer is yes, it does. The major stimulatory neurotransmitter of the brain is the amino acid glutamate, and the major inhibitor is another amino acid, gamma-aminobutyric acid, or GABA. If you would decrease GABA inhibition, then that's a stimulatory effect. So you would give the brains more stimulation and preserve neurogenesis. So they have a couple drugs that have done that, and they found memory and cognitive function restored. This gene has a very strange name, but if you look at the top line, DYRK, it shows you what it means, and the inhibitor is green tea extract. When this gene is present in 3 copies, it will replicate most of the findings of Down syndrome, and you can shut down the bad effects with green tea extract. Okay, and so now I'll mention a few cases that have come to my attention or that I participated in. And so last year, I sort of gave my idea of the biomedical and education before birth and biomedical and education after birth. So I organized these cases along those lines. So the first lady, she was 39 years old. She had a boy with Down syndrome at 40 weeks of gestation. That baby had a AV canal, which is a major cardiac defect, which often occurs in Down syndrome. Before birth, she took high-dose prenatal vitamins. After birth, she breastfed, continuing those vitamins. 80% of the women that have babies with Down syndrome are not able to succeed with breastfeeding, but she made extra effort. She used choline, as seen in one of the studies. And this lady made a vigorous effort to do environmental enrichment for her child. So she read to the baby before birth and after birth, reading flashcards, math, etc. The child was reading at the age of 17 months, understood phonetics at 21 months. Now, this lady would say that she would never test her child. So she doesn't ask the child if she can read, but spontaneously the child at the age of 27 months picked up a kindergarten book and read it cover to cover. At 34 months, the child had a developmental evaluation, and basically the evaluator said, "We've never seen a child with Down syndrome like this." Case 2, this case was one I took care of. She was on high-dose multivitamins before birth. At the time of birth, then, she stopped her prenatal vitamins because she's no longer pregnant. Older research by a guy called Turkel said that if you took high-dose vitamins, the facial features of the children would change and they wouldn't look as much like Down syndrome. So this baby, when it was born, it did not look like Down syndrome. And so when she stopped taking her vitamins, the Down syndrome features reappeared. Last year, when I spoke, I showed one baby that was crawling on the first day of birth. That baby did not have Down syndrome. This baby was also crawling on the third day after birth. It did have Down syndrome, and it crawled very well. The father said that in Down syndrome you have weak muscles and weak tone, but this baby was said to have the strength of a 2-year-old child with Down syndrome. And in general, what I find is that if they take strong vitamins, they'll have very strong muscle tone at the time of birth. And one more, this lady delivered twins at 36 weeks. She took high-dose vitamins before and after birth. Before birth, she exposed the child to music and singing, and after birth, early reading, counting, and sports activities. Exercise is a strong stimulant for neurogenesis. So if, like when I'm preparing this talk, oh my gosh, this is too much effort. I go out, walk 15 minutes. Okay, now I'm doing more on the talk. Those two twins could count to 100 and read and write by the age of 4, and they're very fit, and at adult age, they play all sorts of sports. So, in conclusion, I might say that neurogenesis is a physiologic process. So if you can dial up and down the heart rate or the blood pressure with whatever drugs you do that with, you could also do it with brain development. And so since treatments are coming out with Down syndrome, I think that Down syndrome children are going to be doing a lot better in the future. And if we would stand back and think about the whole society, in our society we have epidemics of autism, learning disability, special education, acting out, violence, and crime are frequently correlated with intelligence. And yet you test these children extensively and you don't find anything. Maybe the cause and the prevention was in pregnancy, and you could test a child until you're blue in the face. You won't find anything. In this country, our prenatal care is becoming dumbed down so it could be done out of a cookbook. And our most important goal for pregnant women is to make sure that they get a mercury-containing flu shot. That's how we work. And, you know, it's as if our developing children are treated like Chinese toys, malfunction and loaded with toxins. In the future, maybe they'll work for the robots instead of controlling them. So what we really need is to consider developing brains as the most important national treasure, and smart, healthy children would be a much better investment than anything else that we could do. Thank you.